Now we will discuss about light and photosynthesis. Light and photosynthesis. So in the previous experiment photosynthesis, we have discussed that carbon dioxide is necessary. Air also involved in it. From the photosynthesis equation, we noticed that in the presence of bright sunlight, so bright sunlight, the plants perform the photosynthesis. What is the relation between the light and photosynthesis? We are going to explain. On this time, the scientists have number of doubts of how the energy is released. When the energy is released, some amount of water and carbon dioxide is released when oxygen combined with carbon and hydrogen. When the oxygen combined with carbon and hydrogen, these are formed. So, and what about the reverse? And what about the reverse? How does the oxygen again is produced and putting back into the air? So from the photosynthesis, oxygen is released as a waste material. We all know very well. So who proved this one? A Dutch scientist, John Ingenhousz. A Dutch scientist called John Ingenhousz proved that oxygen is produced from the photosynthesis. In an experiment with an hydrilla aquatic plant, he noticed that air bubbles are formed around the plant. So the bubbles were named as oxygen. He discovered the bubbles were oxygen. So do you understand that one? So John Ingenhousz in the year 1779, he conducted an experiment. So with an aquatic plant like hydrilla, and he noticed that air bubbles are forming around the hydrilla plant. So he noticed that the air bubbles are oxygen, and he called it as oxygen. So now after him, Enselman, in the 20th century, an Enselman. So Enselman also detect where does the maximum rate of photosynthesis takes place. He detect where does the maximum rate of photosynthesis takes place. For his experiment, he used a strand of algae and he exposed different that algae to different colors of light, which is present in our rainbow. So the algae, when it exposed to different colors of light, gives you a light, violet, blue, indigo, red, like that. So when it exposed to different colors of light, and then he used oxygen sensitive bacteria, he noticed that the oxygen sensitive bacteria is illuminated around the oxygen sensitive bacteria is grouping around this algae when it illuminated by the blue and the red races. So he noticed that the maximum rate of photosynthesis takes place in the colors of blue and red. So in this way, okay, we noticed that one oxygen is also produced from the plants. We understand? John Ingenhousz in the year 1779, he, by conducting an experiment, I will explain that experiment for you now. What is conducted by the John Ingenhousz? So our aim is the aim of the experiment is to prove oxygen is released by the photosynthesis process. Okay, so for this we required a beaker with water, so short stemmed funnel, beaker with water, short stemmed funnel, and also one test tube. short stem funnel and test tube, hydrilla plant. Hydrilla plant. So the aim of the experiment is to prove that oxygen is released during photosynthesis. Okay, now the procedure, I have already told you what are the apparatus. So the second column apparatus I already told you: beaker, water, test tube, short stem funnel and hydrilla plant. Okay, now come to the procedure. Arrange the apparatus as shown in the figure and take some water plants like hydrilla or ellodia. Hydrilla or ellodia. So water plant like hydrilla or ellodia and take small twigs of it and place it in the short stemmed funnel and keep the funnel in the beaker. Okay, next. Okay, na? Keep the funnel in the beaker. Now take a test tube with full of water, invert it on the stem of the mouth of the stem of the funnel. Now arrange the apparatus as shown in the figure. Take some water, hydrilla plants like water plants like hydrilla or ellodia and place it in the short stemmed funnel and invert a test tube with full of water. Invert a test tube with full of water over the stem of the funnel and make two identical sets like this and keep one set in the sunlight and another set in the dark. Okay, I think here we have to notice one thing. The level of the water in the beaker is the level of the water in the beaker is above the level of the short stemmed funnel. Ensure that you have to remind it. The level of the water in the beaker is above the level of short stemmed funnel. So we have taken two identical sets. One we kept in the sunlight and another one in the inside the room. So by noticing that when the setup which is present in the sunlight, we notice that small air bubbles are formed in the place of the water in the test tube. If you notice in the dark room, in the setup view which is present in the dark room, we don't see any type of air bubbles. Now test the gas by taking the test tube outside, by keeping your thumb to the mouth of the test tube and carefully take it outside. By introducing the glowing matchstick or incense stick, we notice that it burns brightly, the flame burns brightly. So we notice that the gas which is useful for the burning is the flame is that is oxygen. So in this way, John Ingenhouse performed this activity. So once again I will explain to prove that oxygen is released from photosynthesis. For this activity, we required all these apparatus. Arrange the apparatus as shown in the figure. Take a water plant like hydrilla or ellodia and keep it in the short stemmed funnel and place the funnel in a beaker. Invert a test tube with full of water over the stem of the funnel and next ensure that you have to remind one thing here. The level of the water in the beaker is above the level of the mouth of the funnel. So in this way, the water will not escape from the test tube. Next, so if you notice that one, the setup which is present in the sunlight, it forms air bubbles. So this if you introduce the glowing matchstick or incense stick, it would burn into flames. So the gas is oxygen. So this is about this one. And also I already told you that Anselman also detected when does the maximum rate of photosynthesis takes place by using a strand of algae, a group of algae, strand of algae, and exposure to the bright different colors of light in the picture. You noticed that it is takes place in the blue and the red races. That means in the blue and red races it takes more amount of photosynthesis. So this is about light and photosynthesis. Now I am going to explain about another important topic, that is structure of the chlorophyll. First of all, who discovered chlorophyll? Where does it located? We will discuss about it. So now I am explaining about the relation between chlorophyll and photosynthesis. So John Ingenuous hypothesized and said like this: all the green parts of the plant perform the photosynthesis. If I hypothesized that in the 1779, on all green parts of plant, all the green parts of the plant perform the photosynthesis. Then on those days the scientists have one type of question. Then what about the plants containing the colorful leaves? So color leaves paristity. What about the plants which contain the colorful leaves? Like example, yellow color leaves, red color leaves. And what about why the plants which first look like in the dark red color and finally converted into green color? What about these plants? How does they perform the photosynthesis? And what about the plant animals which looks like or which are in the green color when they perform photosynthesis or not? This type of questions were there in the minds of the scientists on one days, because John Ingenuous said like this: green parts of the plants only perform the photosynthesis. Then what about the plants that have the colorful leaves? What about the animals which have the green color? So to notice this one, to explain about this almata, after a long time, after 4 decades of the John Ingenuous, the exact location, the scientists extract the location and isolate the chlorophyll. So they isolate the green part in the leaf. After the 4 decades of the John Ingenuous propositions, after the 4 decades, the scientists isolate the green part of the plant. So this was possible by the 2 scientists in the year 1883. Those 2 was Balakoteswararao and Cavendish. So John Ingenuous had a number of questions in his mind. What about the plants? What about the plants containing the colorful leaves? For all these reactions, after the 4 decades of the John Ingenuous propositions, Balakoteswararao and Cavendish discovered one answer. They isolate and extract the location of the green part of the plant, that is chlorophyll, and named that green part as chlorophyll, which means the green leaf. So in the year 1883, Balakoteswararao and Cavendish discovered, isolate and extract the location of the green part and named it as chlorophyll, which means the green leaf. Not only this green one, rather than other colorful pigments also present in the leaf, which involved in the process of photosynthesis. So they isolate it, but no one knows about the exact location of the photosynthesis. Where does the exact location of photosynthesis takes place? Where does the chlorophyll is present? So it was believed that on those days, all the scientists believed that chlorophyll is present in entire cell. Chlorophyll is present in entire cell, but one of the scientists, Julius Van Sachs, 1817, I think, not 1883, 1817. The scientists. So Julius Van Sachs, in the year 1883, he noticed the exact location of the chlorophyll, and he noticed that the chlorophyll is not present in the entire cell of the leaf. It's not present in the entire cell of the leaf. It is present only in a certain cell organelles. So Julius Van Sachs noticed that chlorophyll is not present in the entire cell of the green part. It is present in a cell organelle, and it called that cell organelle as chloroplast. What is that? Chloroplast. So the chlorophyll is present in the chloroplast. There are approximately 40 to 100, number of chloroplasts are present. 140 to 100 may be there. So this is about Julius Van Sachs. 1817, Falter and Cavendish extract and isolate the location of the green colored part and named it as chlorophyll. But the meaning is green leaf. The exact location of the chlorophyll and the rate of photosynthesis. Where does the photosynthesis take place? No one knows about it on those days. So it was discovered by the Julius Van Sachs in the year 1883. The chlorophyll was present in a certain organelle. It is not spread entire the cell. In a cell, number of cell organelles are present. In your 9th class, you have already learnt about cell organelles and its functions. Different types of cell organelles. So the chloroplast is one of the cell organelle. The chlorophyll is present in the chloroplast, one of the cell organelle, and perform the photosynthesis. Okay, the chloroplast is present in the stomatal guard cells. We have discussed that stomata is present in the stomatal guard cells. Guard cells and ground tissues. So ground tissues. So this is about the chlorophyll. Who discovered chlorophyll and chloroplast. Now, on those days, when the scientists try to observe the chlorophyll, when the cell is broken, all the cell organelles also broken. When the leaf is broken, all the cell organelles inside it also broken. But it's not until 1954, a scientist named Daniel I. Arnon was able to break up the plant cells gently, and he noticed the structure of chlorophyll and explained the role of photosynthesis in it. Now I am going to explain about the structure of chloroplast. Dear students, so chlorophyll was discovered, isolated by Falter and Cavendish, 1817. Exact location of chlorophyll is discovered by the Julius Van Sachs in the year 1883. Now, so chloroplast, the structure of the chloroplast. Chloroplast is a membranous structure. So chloroplast is a membranous structure consisting of three membranes. So chloroplast is a membranous structure consisting of three membranes. Okay, the three membranes. The third layer forms stacked. Listen carefully. Chloroplast is a membranous structure. It consists of three membranes. The third layer forms stacked sac-like structure. Stacked sac-like structure called granum. The third layer here, the part is gram. The third layer forms stacked sac-like structures called granum. It is believed to be all enzymatic. It is believed to be trapping of solar energy. So the third layer forms stacked sac-like structures called granum. It is believed to be trapping of solar energy. And the second membrane is fluid portion. Fluid filled portion called stroma. So this is stroma. These are membranes. So the third membrane forms stacked sac-like structures called granum. It is believed to be trapping of solar energy. Solar energy. The fluid filled portion is called stroma. It is believed to be all enzymatic reactions which leading to the formation of glucose and which in turn converts into starch. So this is about structure of chloroplast. Chloroplast is a membranous structure consisting of three membranes. The third membrane forms stacked sac-like structures called granum. It is believed to be trapping of solar energy. The third one, the fluid filled portion is called stroma. It is believed to be trapping. It is believed to all enzymatic reactions. Formation of glucose and leading to the formation of starch. Next, here the substances that present in the chloroplast which capture the sunlight are called photosynthetic pigments. Very important 1 mark question. Photosynthetic pigments. What are photosynthetic pigments? The substances present in the chloroplast which capture the sunlight are called photosynthetic pigments. So there are number of photosynthetic pigments are present in the chloroplast which capture sunlight. The chloroplast is a membranous structure. So chloroplast is a pigment, such a pigment which contains one atom of magnesium. Chloroplast contains one atom of magnesium. So chloroplast is such a pigment contains one atom of magnesium, which is similar to the heme of hemoglobin. In our blood cells, hemoglobin present, which carries the hemoglobin, which carries the oxygen. Like this, chlorophyll also contains one atom of magnesium. Okay, now useful for the performance of the photosynthesis. It is similar to the structure of heme of hemoglobin. So the red substance which contains one atom of iron contains one atom of red pigment. Okay, na? So it is similar to the structure of heme of hemoglobin, which contains the one atom of iron. Okay, so there are two types of major chloroplast. One is chlorophyll A. Chlorophyll A is blue green in color, and another one is chlorophyll B, yellow green in color. So these are the two types of major chloroplast. One is chlorophyll A, blue green in color, chlorophyll B, yellow green in color. This is the structure of chloroplast. Chloroplast is a membranous structure consists of three membranes. The third membrane forms stacked sac-like structures called granum. It is believed to be trapping of solar energy. The fluid portion is called stroma. It is believed to be responsible for all enzymatic reactions which leading to the synthesis of glucose, which in turn forms the starch. Okay, so the photosynthetic substances the compounds, that means the substances that present in the chloroplast, substances which present in the chloroplast captures the sunlight are called photosynthetic pigments. These are called photosynthetic pigments. Chloroplast is such a pigment which contains one atom of magnesium. It is similar to the heme of hemoglobin. Hemoglobin, a red pigment which contains one atom of iron. Okay, red pigment which contains one atom of iron, which is responsible for transportation of gases in our blood. Especially combining with oxygen and formation of hemoglobin. We will discuss it in the next classes. So chloroplast is, there are two types of major chloroplast. One is chloroplast A, blue green in color. Chloroplast B, yellow green in color. Do you understand that one? Okay. Chloroplast, the substances, photosynthetic, the number of substances are involved in it to participate in the photosynthetic process. Okay. So there are several events occurs in the photosynthesis process. What are the events that takes place in the photosynthesis? There are several events occurs in photosynthesis. There are what are the several events that takes place in the photosynthesis? That means in the chloroplast. The events that takes place in the chloroplast is, one is conversion of light energy to chemical energy. Conversion of light to chemical energy. Second one is splitting of water molecule. Splitting of water molecule. And third one is reduction of carbon dioxide to carbohydrates.